Hello, dear friends. Welcome back to my YouTube channel. And today we are going to discuss the structure of SARS-CoV-2, or also what we call it as a novel coronavirus, nCoV. In the previous lecture, we discussed about the what is coronavirus, its classification and its entry, how what are the types of this coronavirus and other things. So today we are going to discuss this structure, their parts, their proteins, and how that virus enters inside the cell. So let's revise. This coronavirus is so named for their crown-like appearance, are a large family of viruses spread from animals to human. The SARS coronavirus is a beta coronavirus which was discovered in one city, Hubei, province of China in December 2019. They are enveloped positive sense single-stranded RNA containing viruses of zoonotic origin. So this all part we have discussed in the previous lecture. Let's start the size. So they are spherical to pleomorphic particles measuring between 80 to 160 nanometer in length. The SARS coronavirus 2 contain four structural proteins, namely the envelope proteins, spike proteins, membrane proteins, and nucleocapsids. So these are the four main structural proteins found in the coronaviruses. So among these, the S protein, that is spike, membrane, and envelope proteins together form the envelope of this virus. The M protein is the most abundant and mostly responsible for the shape of the coronavirus, shape of the envelope of the coronavirus. E protein is the smallest structural protein. Then the S and M proteins also the transmembrane proteins, means they spans the membrane and they involve in the virus assembly during the replication of the virus. The N protein remain associated with RNA forming nucleocapsid inside the envelope. Then, although the N protein is largely involved in processes relating to viral genome, it is also involved in other aspects of coronavirus replication cycle, that is assembly and budding, the host cellular response to viral infection. The polymers of S protein remain embedded in the envelope and giving the crown-like appearance, thus named coronavirus. So the crown-like appearance of the coronavirus is due to the polymers of S proteins. So let's define the structure of coronavirus. As we discussed, the coronavirus structure contains envelope protein, membrane protein, nucleocapsid, and E protein. So apart from these, there are other different proteins which are present in the virus. So this envelope protein is a membrane bound protein. Okay, so this is the membrane of this coronavirus. These are the spike glycoproteins. Okay, those proteins which are present inside or embedded inside the membrane are called as transmembrane proteins. So this is a membrane protein. Then open reading frame is a accessory protein which is also present in the membrane and little bit part outside. So the single stranded RNA virus, we have discussed that. So this is the virus which is covered by the nucleocapsids. So these are small capsomers covers the nucleic acid of the virus, that is single stranded RNA. It also contains the helicase, that is DNA breakdown for. Okay, so when this virus enter inside the human cell, this NSP13 protein breaks the host cell DNA and it replicates its own copy by using NSP9. So NSP9 is a RNA replicates protein present inside the virus, as well as this helicase is also present. Along with this is a NSP15 proteins is there. Then the glycan groups are there. So these glycan groups connects this spike glycoprotein to the membrane, and this spike glycoprotein play important role for the viral entry inside the cell. This spike glycoprotein has one receptor, that is ACE2, human ACE2 receptor present on human epithelial cells, lung cells, or the heart cells, and that human is angiotensin converting to enzyme react in enzyme and spike glycoprotein reaction is carried out. When this spike glycoprotein attaches to the human ACE2 receptor, there are other some receptors, that is transmembrane serine protease TMP RSS2 receptor and other receptors play role for the binding of this receptor interaction, and when this interaction takes place the virus will enter inside the cell. So the researchers are focused on this point, how to prevent this binding. If the binding is not takes place, virus is unable to enter inside the cell. So the research is going on. We are also working on this same process where we want to prevent the entry by preventing this binding. Okay, I'm going to discuss that later on. So the NSP protein is the main protease, that is protein breakdown present inside the virus. Also there is NSP16, that is 2 methyltransferase, then NSP9 10, then NSP33 like papain like proteases. These are the there, and also at the center there are NSP7, NSP8, NSP12, RDRP genes are present. So this is the structure of SARS coronavirus 2 and its virions and its proteins. So this virus, SARS coronavirus, uses ACE2 receptor of the target cell to enter the target cell, inside the target cell. Then SARS coronavirus 2 and SARS coronavirus, which is firstly outbreak in 2003, bind with the similar affinities to ACE2 receptor. The structure of SARS-CoV-2 virus spike glycoproteins in two conformations. Okay, so SARS coronavirus polyclonal antibodies inhibit SARS coronavirus 2 spike mediated entry into the cell. So this is all about the structural features and the entry of coronavirus inside the cell. If you like the video, click the like button, share my video, and